Good evening, one and all. I warmly welcome you to our ninth CellTalk webinar. In today's session, Ms. Soumya Shivarama will be presenting research titled Targeting the Alpha V Integrin TGF-β axis improves natural killer cell function against glioblastoma stem cells. Soumya is a clinical content writer at Renix. She works with multiple teams to produce scientific content such as blogs, scientific summaries, and white papers. She also helps in the collection of relevant clinical and scientific information required for various projects. Soumya has completed her master's degree in science with microbiology from Bangalore University. Before joining Renix, she worked for New Age Systems Private Limited in Singapore and Empower Research, where her activities focused on applied microbiology. Renix has allowed her to not only manage scientific content, but also handle responsibilities shared between various team members. So, without further ado, I now welcome Soumya to take the floor.
Thank you, Aditi. Today I'm going to present on the paper targeting the alpha V integrin and TGF-β axis improves the natural killer cell functions against the glioblastoma stem cells. Original authors are Ila Shaima and others, and the topic is actually based on the immunotherapy against the glioblastoma stem cells. This was published on 2021 in the journal name called the Journal of Clinical Investigation, and the impact factor is 19.46. Let's move on to the general description of the disease, that is glioblastoma multiform. Glioblastoma multiform, or a grade 4 astrocytoma, is the most common and aggressive type of primary brain tumor in adults. It is the most deadly and difficult to treat among all human cancers. These type of tumor invariably reoccurs despite of resection, radiation, chemotherapy, with a median survival time between 14 months to 2 years. One of the significant factors determining this difficulty is the presence of glioblastoma stem cells, which are fundamentally different from the mature cells. These differences include a resistance to standard chemotherapy and radiotherapy, as well as the ability to initiate tumors after treatment and immediate reoccurrence. Coming to the immunotherapy-based cells, that is a natural killer cell-based therapy, is one of the most promising immunotherapeutic strategy for treating glioblastoma. Preclinical studies suggest that natural killer cells can treat glioblastoma stem cells efficiently. These immunomediated attacks are known to protect against tumor initiation, progression of the disease, and metastasis. Most of the tumor cells studied to date have a strong immunodefenses by allowing them to escape from natural killer cells' immediate cytotoxicity. This includes disruption of a receptor-ligand interaction between natural killer cells and tumor cells, and the release of immunosuppressive cytokines into the environment, such as transforming growth factor beta. Present challenges we are facing in these diseases is the rapid growth rate of these aggressive tumors, their molecular heterogeneity, and ability to invade a critical brain structure, and also the regeneration of tumors by glioblastoma stem cells. Objective of this research is to determine how effective natural killer cells are at eliminating glioblastoma stem cells derived from the patients undergoing a primary GBM surgery, and also to investigate the role of both activating and inhibitory receptors in natural killer cells cytotoxicity against glioblastoma stem cells using a receptor-specific blocking antibodies and other inhibitors. There are several methods they have used in this research. The first and foremost is to find out the cytotoxicity assay. For the purpose of this assay is to evaluate the susceptibility of glioblastoma stem cells to natural killer cells mediating killing by using glioblastoma stem cells derived from the patients with the various glioblastoma subtypes, such as a pleural, classical, and also they have used a non-glioma cell line such as U87. And they have kept a K562 as a potential target cells because of their marked sensitivity to NK cell mediating killing. In this experiment, the sample cells are labeled with a 51 chromium. The label is then released from the target cells by cytolysis. The label can be isolated by centrifuging the samples and collecting the supernatant. The supernatant from centrifugation can be counted directly in gamma counter, since chromium is considered as a gamma emitter. As a result, you can see from this graph it shows the sensitivity to healthy donor NK cells, killed glioblastoma stem cells, and a K562 cells with equal efficiency and much more readily than healthy human astrocytocytes, which displayed a relative resistance to NK cell mediated killing. NK cells also efficiently targeted a non-glioblastoma stem cells, glioma cells lines such as a U87. Next, coming to the analysis of expression of NK cell activating or inhibiting receptor ligands on a glioblastoma stem cells by flow cytometry. Actually, glioblastoma stem cells and also healthy astrocytes both contain the HLA class 1 and HLA-E ligands that are inhibitory for NK cells. In this study, receptor-specific blocking antibodies were used to disrupt a specific receptor-ligand interaction in order to access their role in NK cells dependent cytotoxicity against a glioblastoma stem cells. Activated health control natural killer cells were co-cultured with the glioblastoma stem cells in the presence or absence of blocking antibodies. These are the receptors which are only present in the glioblastoma stem cells apart from HLA cells. HLA cells are HLA receptors are present in both glioblastoma stem cells and astrocytes, but NKG2D and DNAM and NKP30 are present only in the glioblastoma stem cells. That's why they have used both anti-HLA stem class 1 and anti-KG2D, anti-DNAM and anti-KP30. And they have tested for a cytotoxicity using a 51 chromium release assay against a glioblastoma stem cells was used for the as I mentioned, cytotoxicity assignment. As you all can see from the graph, the blockade of NKG2D, DNAM1 and NKP3 showing the efficiency in stimulating the NK cell, but not the HLA1 class 1. HLA class 1 significantly decreased the NK cell mediated GSC killing. These findings in NKG2D and DNAM1 and NKP3 possess in the glioblastoma stem cells stimulate a NK cell activation leading to the GCS elimination. Next, moving on to the next test called a NK cells infiltrates GBM tumors, displaying altered phenotype and functions. For this test, they have used a cytometry by time of flight and a panel of 37 antibodies against the inhibitory and activating receptors, as well as the differentiation homing activation markers has been used to gain insights into the phenotype of the glioblastoma multiform tumor infiltrating natural killer cells. And they have also used a heat map analysis to compare the protein expression between the groups. As you all can see from the images, as a result, GP-NK cells and HC-NK cells shows the similar phenotypic expression when it comes to tumor-infiltrated NK cells. it is greatly different from the GP NK cells and the HK cells. This confirmed the differentiation in the phenotype of a tumor initiated infiltrate the natural killer cells. TGF-β1 mediates a natural killer cell dysfunction in GBM tumors. This test is conducted by a co-incubation with the natural killer cells from healthy control donors and a patient-derived glioblastoma stem cells in the presence or absence of a glioblastoma tumor factor, in the presence or absence of a TGF-β1 neutralizing antibodies and accessing their cytotoxicity against control cell and also the glioblastoma stem cells targets. From the graph, you can see the results. TGF-β production by a glioblastoma stem cell contributes significantly to the NK cells dysfunction in the GBM microenvironment. Similarly, but not with the healthy control natural killer cells induced by glioblastoma stem cells. This indicates that natural killer cells are rendered dysfunctional in the suppressive microenvironment of a glioblastoma multiform tumors. Next, moving on to the another test, saying that by disturbing TGF-β1 signaling. In this test, glioblastoma stem cells induce a NK cell dysfunction through release and extracellular activation of TGF-β1. It may be possible to avoid this evasive tactic by inhibiting the TGF-β signaling pathway. Thus, tested with a galinostrib and also TGB receptor 1 kinase inhibitor and a LY210971. The dual inhibitor for TGF-β receptor 1 and 2 can prevent or reverse the glioblastoma stem cells induced NK cell dysfunction. The results are like both neither of the inhibitor affected a NK cell function. Each prevented a glioblastoma stem cells from activating the TGF-β1 and also SMAD2 and 3 signaling pathway in NK cells and inducing dysfunction, thus preventing the natural cytotoxicity of NK cells against control cells and glioblastoma stem cells. Glioblastoma stem cells induce natural killer cells dysfunctions through cell-to-cell contact dependent on a TGF-β release. To prove this, they conduct the transwelt experiments with the donor-derived natural killer cells and also glioblastoma stem cells were either directly in contact with each other or separated by a membrane with a pores of 0.4 mm, which allowed a soluble molecules to diffuse but not the cells. Direct contact of glioblastoma stem cells with the natural killer cells resulted in significantly higher level of glioblastoma β1 compared with those attained when a glioblastoma stem cells were separated from natural killer cells by a transwell membrane. This indicates that release and activation of a TGF-β1 by a glioblastoma stem cell is a dynamic process requiring a direct cell-to-cell contact between a NK cells and also glioblastoma stem cells. Alpha V integrin mediates cell contact dependent TGF-β1 release by a glioblastoma stem cells. The alpha V integrin is a heterodermic complexes of alpha V beta 3, alpha V beta 5, and alpha V beta 8 are highly expressed in a glioblastoma multiform, particularly on a glioblastoma stem cells. This study is used a small molecule inhibitor called a selignitide, which has a high affinity for a alpha V integrins, alpha V, and which prevents a glioblastoma stem cells induced NK cell dysfunction by lowering a TGF-β1 production in the supernatant in natural killer cells in direct contact with the glioblastoma stem cells. And also there is one more method to prove that alpha integrin initiated TGF-β1. That is called a genetic slicing of pan-alpha integrin in glioblastoma stem cells using Cas9 gene editing. CD9 and CD103 are upregulated on GBM-TI tumor infiltrate natural killer cells and can be induced on healthy natural killer cells after co-culture with the TGF-β1. The CRISPR-Cas9 gene editing technique was used to knock out an CD9 and CD103 in healthy donor natural killer cells and tested with the cytotoxicity of a wild type. Comparatively to wild type controls, slicing CD9 or CD103 improved the cytotoxicity functions of natural killer cells when co-cultured with the glioblastoma stem cells. Conversely, CD9 and CD103 double knocked natural killer cells co-cultured with the glioblastoma stem cells retain their toxicity against the control cells. This suggests that alpha V integrins on a glioblastoma stem cells bind CD9 and CD103 on natural killer cells to regulate the TGF-β1 axis involved in the TGS-induced NK cells dysfunctions. Next, coming to the in vivo experiment that is called the inhibition of alpha V integrin and the TGF-β1 axis enhances the natural killer anti-tumor activity. In this experiment, they have used a 2BDX mouse model of patient-derived glioblastoma stem cells in which it was implanted on the right forebrain of mice, and alpha V integrin and TGF-β axis was interrupted using an alpha V integrin inhibitor, a TGF-β receptor kinase inhibitor, or by genetic disruption of TGF-br2 using CRISPR or Cas9 gene editing. The combination of NK cells with either gallium-nitride or to block the TGF-B signaling or a selenide to block the integrin pathway improves the anti-tumor response. The tumor bioluminescence used as a surrogate to access the tumor progression rapidly increased in untreated mice than in the mice that were treated with the natural killer cells either alone or combined with the selenide or a glanistrib led to a significant improvements in a tumor control. The best overall survival was observed when the mice received NK cells combined with a gallium-nitride or with a selenide compared with the untreated controls. TGF-br KO NK cells treated with mice also resulted in significant improvement in the overall survival when compared with other untreated controls. Coming to the discussion part, they have confirmed a few points what they observed in the methods and results. The TGF is abundantly present in the GBM microenvironment and is released by the tumor as well as several other cell types such as the regulatory T cells, macrophages, myeloid-derived suppressor cells. NK cells and these glioblastoma stem cells cell-to-cell contact requires the interaction between alpha V integrins on glioblastoma stem cells and CD9 and CD103 on NK cells. TGF-β1 is released by glioblastoma GBM cells and is irreversibly suppress the cytotoxicity functions of natural killer cells by inducing changes in their phenotype, transcription factors, cytotoxic molecules, and chemokines. An important aspect of this studies is the interaction between the alpha V integrins on these glioblastoma stem cells and the TGF-β1 induced receptors CD9, CD and CD103 on NK cells, acting as the main mediators of TGF-β1 production and subsequent NK cell dysfunction. It is confirmed that silencing the alpha V integrin action in glioblastoma stem cells by a CRISPR and a Cas9 gene editing or a pharmacological inhibition with a selenide prevented a glioblastoma stem cell induced natural killer cell dysfunction and decreased TGF-β1 production. This combinational approach of a natural killer cells immunotherapy with the TGF-β 1 and the alpha V integrin inhibitors such as a selenitide to block a TGF-β 1 signaling by the glioblastoma stem cells. Alternatively, gene editing strategies to delete a TGF-β2 in NK cells could be used to protect against the TGF-β 1 binding and consequent immunosuppression. As a conclusion, on the strength of these findings, the development of an immunotherapeutic strategy utilizing the NK cells derived from healthy donors combined with alpha V integrin inhibitors or genetically edited to silence a TGF-β2 in order to protect them from immunosuppression, which could enable them to recognize and eliminate the tumor cells with the stem-like properties such as the glioblastoma stem cells. The major impact of this research is actually immunotherapy is a promising treatment for patients with a glioblastoma, but it had a limited success so far. Genetically modified natural killer cells allowed us to overcome the immunosuppressive environment in the brain, which allowed us to eliminate tumor regenerating glioblastoma stem cells. Natural killer cell therapies have been showing a promising results in earlier trials, and similar strategies could be applied to other solid tumor types. Thank you.
Thank you so much, Soumya. That was truly an illuminating and educational talk. I'm sure we've had a lot of takeaways from this webinar. I will now open the floor to the audience for any questions, concerns, or opinions that they have for Soumya. I see a few questions in the chat box, so I'll read them out. The first question is from Nupur. What is the role of blocking antibodies, anti-DNAM and others?
Antibodies and
anti-DNAM.
Anti-DNAM. Anti-DNAM is the receptors on the natural killer cells. So, it is a antibody, but in 1 second, I'll go to the slide, so it will be easy for me to explain.
There is a plot of percentage of cells killing where anti-DNAM.
So that is the anti you mentioned that receptor name, right? That is present only on the GBS glioblastoma stem cells.
Yes.
So that if they block that receptors,
Okay.
so
Glioblastoma
the cytotoxicity functions will be
the glioblastoma stem cells. Yeah.
Yes, exactly. So if they block the receptors, the functions will be stopped. The cytotoxicity functions will be dysfunctions will be avoided. Dysfunctions of a natural killer cells will be avoided.
Okay.
Yeah.
Thank you.
I hope that answers your question, Nupur.
Yeah,
the next question is from Vijay. He asks, what are the clinical applications of this research for routine treatment of glioblastoma patients?
Actually, so far they have done the only preclinical test. As I mentioned, this is just a prediction. You can use it for the glioblastoma to stop initiation of the tumor and also stop the initiation, progression, and the metastasis of the glioblastoma in the earlier stage itself, or else reoccurrence after a primary treatment, chemotherapy, radiation, or a resection.
I hope that answers your question, Vijay. So I don't see any more questions in the chat box, nor are there any more questions in the question answer panel. So thank you so much again, Soumya. So while closing this webinar, I would also like to inform the audience that the next CellTalk webinar will be held on the 26th of August 2022, and Ms. Kirti Reddy will be presenting on the topic integrating digital pathology and mathematical modeling to predict spatial biomarker dynamics in cancer immunotherapy. Thank you so much for your attendance and for your presence in this webinar. And I hope to see you next week this time again. Thank you and have a nice day.